Was that you?
Yeah, it's okay. I had my door open. I didn't think to close it first. Sorry, guys. Is this showing my screen when I do this? I don't know if you guys can see it. Because my iPad's been having a problem with this.
Yes.
Okay, awesome.
Screen.
Okay, yeah, call me out if it doesn't stay, because I don't always know. All right. Do you guys have any specific questions that you want me to go over? Or do you just want me to like pick some randomly?
I kind of went over your unit 4 in the meta just because it's new. So I was like, well, I'm gonna go over that more this morning. I don't know. So on I guess basically know everything about like the pathogen, what causes it, how it would be treated, signs and symptoms, all that.
Yeah, if it's on the study sheet, right? And so if it's not on my study sheet, then you don't need to know it, like quite literally. I mean that.
Yeah,
So
it gets very broad. So I'm like, okay, let me get back to the study sheet. I don't really have anything in specific.
let me plug this in. So let me see if I can open what I want to do. Do I have my questions on here? We do not. Let me see. Close this. This is way far down. It's this one. All right. Lecture. Let's see.
So I do have a question. How I'm trying to figure out the best way to make a chart for these, like without it being too much, too busy. You know?
So one thing that you can do, I mean, if you're going to do a chart, I mean, I guess approaching it with a chart is fine. Let me pull up all the stuff I'm trying to pull up, and then I always like change my mind on the thing that I want to pull up. Import first this.
Or what is the best way? I was just saying that it says create a chart on your meta question papers, you know?
Right. So do it how you feel is appropriate. If I was going to be making a chart, then I would probably be looking at my unit 3. So let me see. Let me twist that. Files. I know I have this one. Work. Lecture.
I'm thinking like for the unit 4 like diseases. That's what I was kind of.
Yeah, okay, that's yeah,
Okay.
we can look at that too. So no, I'm not gonna have that on here, I don't think. Yeah, so that would be unit. I don't know why I was looking at unit 3. Of course, it would be unit 4. I'm trying to think the best way to open this would be, I think it would be like this. Okay, here. So we'll do modules. It's going to take forever. We're going to week 16. All right. So if I was going through this, I would make a list of all the diseases. here. I'm not going to go through the whole thing, but if we're talking about the diseases specifically, so not necessarily the causative agents yet. I would first make a list of my diseases. So we're starting off with impetigo, necrotizing fasciitis. We have scarlet fever. We have rheumatic fever and strep throat. We also have another impetigo. So I'm just already listed it once. I'm not going to list it again. We have MRSA, kind of a disease, but this is really more of a species. But I would give it its own card on the diseases. And then I would, where are we at? Gas gangrene, chickenpox. So I'm just going to list until I run out of room on this view here. Then we have smallpox. All right. And so I would start with my list like that. And then I would make a card for each disease. So I mean this, like for each disease specifically, I would make a card. And on the back of that card, the disease, I would have cards. On the back of impetigo, I would say that it can be caused by Streptococcus pyogenes. I would say that it's caused by Staphylococcus aureus. I would also say that these are caused by bacteria. What happened? There it goes. As far as that, if I needed to look up any more about that, we know that this is a skin disease, the skin disease chapter, right? And this was the one that had the flakiness that was associated with this. Exfoliation. Just the basics to describe the basics of this stuff. That's it. That's what I'd have on the back of impetigo. I would do this for every single disease, whatever the basics were that I need to know about it. And if there's any other info about this, like for smallpox, for example, I haven't just said that smallpox is, a skin disease. We know that, but we know it's also characterized by a fever, malaise, and a disfiguring rash here. Yes, give me the thing. Close. Okay, well, never mind. I guess that doesn't work the way I thought it did. So you would want to have that on the back of smallpox. You would also probably want to put that it was eradicated due to the first vaccine. Vaccine was made by cowpox. So I put that on the back of my smallpox card. But I would also make cards of the actual agents, the pathogens. So if we started from the top, we could do that. We have Streptococcus pyogenes. I would have a card for that. And on the back of that one, just write it down below it because it would be a lot. But on the back of that one, I would say it was a bacteria. For sure, I would know that. I would also say that it causes all of those diseases. I'm not going to write them all out, but you get the idea. So that's the bare minimum of what I would do to prepare for this stuff. This is how I would start preparing for this information. And again, base it off of what is actually on that study sheet. If you need a little bit more information, like you should be able to infer from the study sheet that, impetigo is a skin disease because it's in our skin and eye disease chapter. But if you want to have any more information about it, like it's a flaky skin disease or whatever, include just the basics of what we covered. You don't need to go into the exfoliative toxins. You don't need to go into like the beta hemolytic or alpha hemolytic or anything like that, or the bacitracin, nothing like that. It's not on the study sheet. Those are too many details. I know we covered all that stuff, but it will not be on the exam. If it's not on here, it won't be on here, but you do need to know at least the basics. And I would say that would be the bare minimum of what I would understand for the basics. What the disease is, the pathogen that causes the disease, and what type of pathogen it is, as well as its name. That'll be my bare minimum. If I have any other thing written on the study sheet, then I'll add that in too. So where I specifically say that smallpox causes a disfiguring rash, that should go on a thing, right? That's not the best example, but like some of these guys that I talk in a little bit more detail about, you know, where I say that bubonic is infections in the lymph nodes. You can't just know that plague causes bubonic plague. Know that a bubo is an infection of the lymph node, that sort of stuff. So if it's on the sheet, you have to know it. That's what I would do to start with making my notes for that unit. Then I would, after making that unit, try to answer the questions in the meta stuff. So it says create a chart of the different diseases of the skin and eyes and include pathogens that cause that disease, what major group that pathogen falls into, and any key characteristics. And this is literally what I'm talking about. This is how I would start with. That is literally just that. Just that. The diseases, the pathogens, and then the major group, and anything key characteristics that is in the study sheet. So that's be where I would start all of that. Because this unit, even though we cover a lot of stuff in it, this unit is going to be used for the other questions. So on the exam, when I ask you a question on the exam of, you are doing a gram stain, and the gram stain shows, you know, gram-negative diplococci. So let me pull this thing up here. So in chapter 20. So if I tell you that my gram stain shows gram-negative diplococci, that is going to be causing a petechial rash associated with meningitis, which of the following categories does it fall into? Then you should know that it's bacterial. And the reason that you would know that is because our meningitis bacteria, we have Neisseria meningitidis, which is the meningococcus, which is transmitted through close contact and causes the petechial rash. So that's where making that chart will help you with, is when I ask you questions like that. If I don't give you like, is a Neisseria meningitidis, is it a bacteria? Or which of the following are causes of meningitis? And questions like that. Those actually will be on the test, but I'm also going to expect you guys to do a little bit more understanding beyond that. So when I'm asking questions about actual disease presentation or production of antibody or anything like that in the context of how the body is behaving to these. And so one of the things that we talked about in my class right before you guys in our discussion was we basically discussed HIV. And so the fact that HIV, let me pull up another sheet here. All right. HIV virus might float around outside in your bloodstream. And then as it passes through the lymph node, when it's getting circulated back through the lymph, then you might have a B cell that has a receptor on its surface, which we talked about. It's going to be IgD. Then this will see the outside thing, the HIV spike protein, let's say. It'll take it inside. Because it came from outside, though, it'll get presented on MHC2. Just put a 2, just to make it very clear. That's antigen. We know this is a T cell because it's getting presented on MHC. We know even further than that because it's MHC, the molecule that sees MHC2, because it's MHC2, the molecule that sees, specifically, this is what the molecule does, is it sees MHC2. This is CD4. If it is CD4, it is a helper T cell. And then this CD4 helper T cell will make a couple of effector cells, TH2 and TH1, still CD4 positive. And then these guys will come back over here and activate the B cell, which will then make a plasma cell, which produces antibody. First IgM, and then it'll switch to IgG because we've got bloodstream infection. We'll prefer making IgG. So whenever you test the blood of somebody who has HIV, even if they don't have the virus in their body, they will have IgG because this process happened. But it's more complicated than that. Because what cells do HIV infect? Do you guys remember what cells HIV infects? Right? Mm-hmm. So there's a problem here because what will happen is you will have a CD4 positive T cell. This could be any T cell. It could even be this one up here. Or maybe this one has a T cell receptor that sees MHC2. It's a CD4, so it's MHC2. Presenting antigen. Maybe this one sees antigen that's associated with, I mean, I don't even know. It could be HIV. Get this out of the way really quick. Whatever. Oh, never mind. Okay. Let's make this small. Move this. All right. So maybe this CD4 T cell sees MHC2 being presented. It doesn't matter. It's an antigen presenting cell that presents it. But this antigen might be to anything. But any CD4 positive T cell can then become infected on the inside with HIV. So if CD4 T cells, now this is very important, though you understand this. If a CD4 positive T cell, maybe this is a T cell that responds to the plague, I don't know, and is able to present and is able to react to antigen being presented on MHC2, that is plague in this case. But it's infected inside with a whole other thing, which is HIV. So it's infected inside with HIV. How does the CD4 positive T cell call for help? It's still a nucleated cell. It is still a nucleated cell. It is, remember, because it's an A granulocyte. It's still a cell. It's still got a nucleus. It's still got a nucleus. So a nucleated cell that is infected inside with HIV. Is it gonna call for help? Can it call for help? It is, and it will. It'll call for help on MHC1. It's a nucleated cell. It's infected. It will present the HIV antigen on MHC1. Then, oh my God, with this is annoying. Just at the top. Here we go. So here's the HIV antigen. Then we know this is a T cell. We've got antigen being presented by a CD4 positive T cell on MHC1. What T cell is going to respond to this? This is a CD8 positive cytotoxic T cell. And then our T helper cell that we made just right above it to HIV. We had it present on MHC2 of our B cell. CD4 of our T helper cell got activated. We made a Th2 and a Th1 response. This Th1 against HIV will then activate our CD8 positive T cell. And then that T cell can go out and kill any CD4 positive T cell that has been infected with HIV. It'll kill any cell that's been infected with HIV that is presenting the HIV antigen on MHC1. It just happens to be that HIV infects CD4 positive T cells. And so any CD4 positive T cell that is infected with HIV will call for help by posting the HIV antigen on MHC1 and then activate the CD8 T cell response, because the T helper cell that already saw HIV, or the T helper 1 cell that saw the HIV earlier, can then send that signal to activate that CD8 positive T cell to kill any infected, any HIV that's on MHC1 in infected cells. That includes your entire CD4 T cell population that's now infected with HIV. This is why your T cell count drops with HIV. HIV itself doesn't kill the cells. Your CD8 positive T cells kill your cells. This is the level of meta thinking that I expect you guys to be able to pull from this. Now, I went through this and I talked this through with you guys, but you should understand the immune response well enough to know why then our CD4 positive T cell count. If I were to tell you HIV doesn't kill your CD4 T cells, it doesn't. So why do they go down? Because you'll have a CD8 positive T cell response against any infected cells. And HIV only infects CD4 positive T cells. So you're going to be killing any infected CD4 positive T cells. That's how you lose your captains. And that's what leads to the opportunistic infections associated with HIV. So that was one that took it to a little higher of a level. But I'm not just thinking about this, right? Because I'm also thinking about the fact that whenever you get HIV will get into the cell. By docking with, molecules on the surface. In this case, it literally docks with CD4. So it attaches to CD4, and then the T cell gets infected. The HIV moves into the cell. And then we get the mRNA. Well, it's not mRNA, it's just RNA genome that will then get converted into DNA. So this is by a process that we call reverse transcription. Driven by the enzyme reverse transcriptase. And so now we've got a DNA version, and we've got the host DNA. And then we will insert the viral DNA. I'll insert the viral DNA in here. There's a special term for the viral DNA when this happens. This is going to be the provirus. Then that provirus will then make mRNA through transcription, where our RNA polymerase will bind at the promoter of the DNA, split, and the DNA will get split apart. And we will make that exact copy, where if the DNA was ATTCG, the mRNA made from that of our virus, provirus, would then be UAAGC. Transcription, and that will make the mRNA. Then the mRNA will get shuttled out into the cytoplasm, where you'll have your endoplasmic reticulum. These are your ribosomes on the rough ER. And then our mRNA from our virus will get translated into protein. In the rough ER, that'll happen. In the smooth ER, we're going to be adding on our glycoprotein, our glyco, which is our sugars, to make the actual glycoproteins that are the spikes of these guys. And this is going to move through the Golgi apparatus and get shipped this is the Golgi, which will then ship it to the surface of the cell, where we can then stud the surface of the cell with viral spikes. In the meantime, we will also make proteins that we will translate out. That'll be capsids. And from this mRNA in here, we can use this as the genome. The capsid plus the genome is known as the nucleocapsid, which, you know, has a nice happy little shape to it that we call icosahedral. And that this is going to push its way out through those spike proteins on the surface and then coat itself in a little envelope and package itself up with all these spikes on the surface. So that's how that virus is going to be replicating. Well, I explained that to you like this, but I need you to be able to understand that, okay, if HIV is inserting itself in our genome, by what process are we going to get the spikes how does that get spikes on the new viruses that are made? How do we get the spikes there? So you need to be able to tell me that, oh, okay, well, we'd have to go through transcription and translation. We'd stud the surface of our membrane with these proteins, and the nucleocapsid will bud out of it, based on everything that we've learned through units 1 through whatever. Right? So this is where these meta questions, I feel like, are going to kind of come in handy. And if we start looking at some of these and trying to break them down a little bit, what I want to do is whatever, sure. Just have them here like this. Okay. Make this big again. So if we start talking about some of these guys, let's pick one. Well, of course, I put the page here. Let's put it after the meta. Well, put it after, no, forget it. Anyways, let's just pick one. Okay, compare and contrast inducible. Oh, this is a good one because you guys suck at operons too. Inducible and repressible operons and how they're controlled at the genetic level. All righty. Well, an operon is a set of genes only in bacteria. Operated by a single operator region and promoter region. This means that at the DNA level, we've got the promoter, we've got the operator, and then we've got genes. Gene 1, gene 2, and gene 3. And that they're all going to be controlled by that one promoter and operator region. All righty. So in an inducible operon, we start off as off. This means basically this is the DNA, but this is what on would look like. This is at the DNA level. This is what on would look like, and this is what off looks like, no matter what kind of operon we're talking about. All righty. So I gotta actually draw the part where it's off. Let's see. Why did I do that? This is the repressor. Inducible operons start at the off. Inducibles will start here and then turn on by removing the repressor. This means that the inducer binds to the repressor, causing the repressor to change shape. And then fall off. Now, as a result of that, RNA polymerase can bind at the promoter and can transcribe. So you guys remember what transcription is, right? I know I just mentioned it, but just to remind you guys, transcription is whenever we make RNA from the DNA template, right? So now we can take the DNA, because this is at the DNA level. Now we can bind our RNA polymerase, which will bind to the promoter, and it will be able to move through and make the products. The repressor blocks that from happening. So our RNA polymerase would bind and it would not be able to move forward because the repressor is in the way. And that's that. So that's the inducible one. And that was way darker than I meant for it to be. So the other one is the repressible. This one starts at on, meaning there's no repressor bound. so it can turn off. So when the repressor adds to excess product, if the body isn't using up whatever is made in the downstream from our RNA polymerase, this stuff down here, if the body isn't using that up, it'll build up and then will bind with that repressor. Repressor changes shape. And then binds. It now binds to the operator, blocking and stopping RNA polymerase, and thus the production of the genes that are downstream from it. Okay, so I think we covered that question pretty well. I hope that I did a pretty good job on it, and maybe I covered some stuff that you guys wouldn't have. There's one other thing that I want to talk about that I went over from my other class that I'm just now remembering that I don't want to leave you guys out of benefiting from either. And that is, let's say that we give you a scenario, and it is, bigger. You use scenario, and in this scenario, we have at the Oklahoma State Fair, we have now come down with 800 cases of typhoid fever. First of all, typhoid fever is a more serious type of salmonella. What type of pathogen is salmonella? It is bacteria. So, in this particular type of typhoid fever, which is a diarrheal disease, if we take a sample of our patient's diarrhea and just if we were able to just isolate our pathogen, a method that we could use to do that might be our quad streak plate, right? And then picking out individual colonies based on that. If we were able to isolate that pathogen in the culture somehow, and we notice that the gram in a gram stain, we see cells that lack mitochondria. Is this in alignment with our diagnosis of salmonella? If we see cells that don't have mitochondria, would that be a correct result if our patients were diagnosed with salmonella? Yes, this is correct. Because these are bacteria, and bacteria don't have mitochondria. All right, so we've got this. We've got 800 people with typhoid fever over the course of the entire week of the fair. So we have collected a large sampling of people that just attended the fair altogether. So all of our attendees, they either have typhoid fever, they're either positive for the disease, or we have attendees that never got the disease. We also took all of these infected people versus our uninfected people, and we asked them if they ate at the funnel cake place, whether they ate at the chicken place, the Indian taco place, or the hot dog stand. You might already have your suspected results of what you might think is going on. But as it turns out, of the people that have the disease, 700 of them went to the funnel cake place, 800 went to the chicken place, I don't know, 20 of them went to the Indian taco place and 100 of them went to the hot dog place. Of the people that don't have the disease, we have 5,000 that went to the funnel cake place, 0 that went to the chicken place. I don't know, another 5,000 that went to Indian tacos, and then 1,000 that went to the hot dog place. Based on this, we would suspect that the salmonella originated from the chicken place, right? All right, so this is pretty good correlation. So what type of epidemiological study did we just do? This is a case control. This particular outbreak, is it spreading from person to person from this initial incident? It might be at home through fecal oral transmission, but it will probably be self-limiting once we have nipped this particular cause in the bud. So this is more likely to be a sporadic incident and not necessarily epidemic, because it will eventually be stopped from person to person transmission. And it is also going to be a particular type of like spread source. Now, our basically we could have sources that were like a point source or a common source or a propagated epidemic. And so this, I would say, is a common source, not a point. A point source would be one particular thing at a particular instant in time. I walk into the classroom and I sneeze, and then people get COVID after that, then that would be a point source. We knew that this came from this one sneeze. In this case, we have over an entire week, people getting sick over time. It is from a common source, though. So this is a common source. Point source will be happening just from one exposure instant. So these are all different kinds of pieces of chicken when we're talking about 800 different people served here. So this is going to be common source. All right. So that was one thing that we did talk about in my previous class that I didn't want you guys to miss out on. Is there anything else that you guys want me to go over? Do you want me to give you a protozoa disease or anything like that? We can try doing that too. Let me look at this, make sure that I'm still getting, see if you guys, because I can't see if you guys chat on this, I won't be able to see it. No chats. Yeah, to make sure I'm not missing anything. All right, so let's go through a situation where somebody has diarrhea and they were swimming in a pool. It's a chlorinated public pool. And I'm going to tell you that there is a particular disease which has these hardy cysts versus trophozoites, all right, that are going to be resistant to chlorine. The trophozoite versions of these guys are the disease-causing multiplying version. Anytime you have disease being presented, the version of that protozoa that's causing the disease is always a trophozoite. The cyst is the one that's the resistant, and it is completely dormant. It is inert, just like our endospores are for our bacteria. So this is going to be a protozoa. In this case, Which of the following organisms could it be? We've got diarrhea. We've got these cysts in the trophozoites. Is this Clostridium difficile or Clostridioides difficile, however you want to write it? Salmonella, S-tech, or Cryptosporidium. Any guesses? Any takers? It's diarrhea. We have identified that there are cysts and trophozoites. It's in a public pool. Therefore, if we know there's cysts and trophozoites, this has to be a protozoa because that's a defining characteristic of protozoa. So the real question here is, which of these are protozoa? D. This is protozoa. E. coli here that produces Shiga toxin and causes dysentery. Salmonella enterica, this is a bacteria. Oh yeah, and E. coli, just to be clear, is a bacteria. Clostridioides difficile, also a cause of diarrhea associated with pseudomembranous colitis. This is a bacteria. It's endospore forming. But it is still a bacteria. All right, so with this guy, we know it's a protozoa. Now, if I look at the cysts under a microscope, and now we see mitochondria here. Is this consistent with Cryptosporidium? And the answer here is yes, because Cryptosporidium is a protozoa. Protozoa are protista, which is a kingdom of eukaryotes. True nucleus and true organelles for that. Okay. All righty, let me see. I'm trying to think of, we kind of went over the MHC stuff already. I could do phagocytosis and stuff. I feel like we'd be better spent trying to talk through this stuff. But let's do the vaccines. Let's do this. Explain in as much detail as possible all of the metabolic, genetic, and immunological factors involved in how the body reacts when inoculated with the mRNA COVID vaccine. All right. So basically for the mRNA COVID vaccine, first, the mRNA enters your cells through the injection. Do we need to transcribe it? No, because we already have mRNA. You'll go straight to translation. So go ahead and translate that mRNA, and translation will mean we're taking the mRNA and using ribosomes made up of rRNA and tRNA to read the codons. And that's going to make us COVID spike protein. Now your cells will be able to elicit an immune response. So this will be something that's made from our mRNA, but that we can put on the surface of our cells. So spike protein on our cell surface, because that's where the normal COVID spike protein would go. Because remember how we talked about with HIV, whenever you make a virus, you'll get your capsid, and the capsid will bud out of your membrane, your cell membrane, that is studded with the spikes, right? This is true for HIV and for COVID. So those spikes naturally want to be on your cell membrane. So that's exactly where they're going to go. So your spike proteins on your cell surface membrane cause an outside response from your immune system. So what this will mean is we might have a macrophage that will see those surface spike proteins and be able to ingest it. And present it on MHC2. This is going to be seen by a T cell. This CD4 positive, because we're seeing MHC2, which we already talked about this, so I'm not going to spend too much more time on this. This is going to activate these. Then this guy is going to make TH2 and TH1. But we don't have any inside to react to. So this guy won't have really much of a job to do. But our B cells that are capable of seeing that antigen with their IgD receptor can see COVID spike protein and then present it on the MHC2 and whatever, and then the TH2 cells can then activate that, and then these guys in turn will become plasma cells that then make antibody against COVID. Now, taking this further, looking at this right here, what's really going on when we activate our CD4 positive T cell? First, we have to proliferate out that clone, the anti-COVID spike clone. Then it'll be able to then it'll be able to differentiate. So first we have to proliferate, and this means to make copies of that cell. So in order to make those cell copies, we need energy in the form of ATP, which if we are proliferating and differentiating our CD4 positive eukaryotic T cells, this ATP production happens in the mitochondria of those via aerobic respiration. Then we can use that ATP to fuel the DNA replication and building our new cell parts. Building new cell parts is going to be an anabolic process, and that requires the input of energy in the form of ATP as well. So we'll need ATP for that too. You'll also, because we're building new parts, some of those parts are going to be lipids, some of those parts will be carbohydrates. That'll all be made in the smooth endoplasmic reticulum. And any of the proteins that we're putting on the surface of our cell, if we're duplicating our CD4 positive T cell, one of the proteins we already need to make is going to be CD4 itself, as well as our T cell receptor on our T helper cells that we are proliferating. So those are two proteins right there that we'll need to make, let alone making copies of our DNA polymerase and RNA polymerase themselves. All of that is protein production, and that's translation. And then we're going to have to first transcribe from the DNA genes that code for those things. Anyways, that's how we're going to be leading to building of our new cell parts. And so we see here that asking the question about the mRNA vaccine and all possible how the body reacts to that. At the immune level, we can do that, but then we can also do it at the biological level, talking about how we're going to be making all those products to begin with. All right, so that's the kind of level of explanation that I'm looking for in these meta questions. Literally tell me absolutely everything that you can think of and wherever that might lead you in the discussion. And wherever you get stuck, that will unearth the things that you don't understand as well as other things, right? Okay, and so, whoops. All right, so one of the things that it also asks with that is include explanations for perceived side effects, in addition to the basic cellular responses and immune responses. So we went over the immune responses, but what other side effects might we have? Okay, so here's some stuff that we can think through. What you're going to have are cells that are basically studying themselves with COVID spike protein. The immune response is going to be able to react to that by making those antibodies that we're eliciting. That's the whole point, to create the memory for that. But the initial response is we could have antibody sees that foreign thing. We could also have complement that could see it. We could also have overall inflammatory response to this. This is something that is seen as foreign. And so this could initially just trigger our inflammatory response to that. But one of the things that might happen is once you make your antibody, seeing that foreign thing, if that mRNA is still expressing that on the surface, as this is just getting started with our response here, we could also have our inside response. So we have also COVID on the inside being expressed out on MHC1, and that would be activating our CD8 cells. Here's CD8 T cell. And then we would have our T helper cells from up here, potentially. This isn't as important for the memory, but can induce the initial side effects that we see. And then this can then kill with those granzymes and perforins. So then you will start having an inflammatory and cell destructive response while these cells are making this foreign protein that we haven't ever been trained to treat as a self protein. And so your body can start attacking those cells. However, the mRNA is short-lived. And so this will be a self-limiting reaction. We don't have a microbe that is multiplying and causing us to continuously make this. We're not going to have a continuous reaction to it. It'll be easily taken care of by our inflammatory and cell destructive responses. So this is a self-limiting thing. And so this is where you get COVID arm. I'll be coming from that. So that inflammatory response, as well as the CD8 cells, antibody basically activating complement and leading to the inflammatory response and all that. But again, short-lived. Most of those cells will be gotten rid of very quickly, and not every cell in your body is expressing this, but the ones that are can potentially be attacked this way, and then that will lead to inflammation in the site. Again, it's not going to be deep penetrating expression of this thing. It's only going to be a superficial and a few of our target cells that got affected by this. Because we're not multiplying out that mRNA. We're not making copies of the mRNA. We're only expressing it in a few of our cells. All right. Come on, thing. Keep up. So let's come back up to the questions. Our patient has a skin infection. What questions might you ask the patient about environmental or lifestyle factors to better guide your diagnosis? Have you had any hospital stays recently? Have you handled any outdoor stuff? Anything like that. So let's say that our patient works on a farm and they recently cycled their livestock off of a field, and now they're tilling an old livestock field. All right, so we are also taking a look at our skin lesion. We would also be able to get more information from that. Moving forward just from this, now, if they got this from an old livestock field, I would be concerned about bacteria that form endospores. In particular, because this is a skin infection and is exposed to the air, I'm not concerned about anaerobic species. It's not going to be anaerobic because it's on the skin. It's exposed to the air. So I'm going to be more concerned about other endospore forming species. And since Clostridium are all anaerobic, this can only be Bacillus species. And one of our endospore forming species that is found as zoonoses in farms might be Bacillus anthracis. So what lab tests might I perform? I might be able to identify the pathogen just based on the sore. And so what kind of sore would indicate cutaneous Bacillus anthracis? That would be a black eschar. So that could help me diagnose it. Maybe one thing that I could do to diagnose it would be to perform a PCR test. In that case, I would need primers for genes that are specific to B. anthracis. Also, if I think this is B. Bacillus anthracis, this is a skin infection. And well, I think it's specifically this. This is another thing to remember on this time is this is a bacteria. And if it's endospore forming, I could do PCR. So it started there. And another one I could do is an endospore stain. And a gram stain. Now, the problem is that in our patient's actual skin wound, we have vegetative bacteria. If those vegetative bacteria in the skin wound aren't forming endospores, our endospore stain might not be positive. So we might want to culture it and let it sit for a few days and create a stressful environment for them, and then see if it forms endospores, or we could just go ahead with a PCR test if we have the primers for that, or we could perform a gram stain as well. So if it was an Bacillus anthracis and it does make endospores, then what kind of gram stain would it have? Purple for positive, because all endospore forming bacteria are gram positive. And so these are some tests that I could do: the endospore stain, the gram stain, maybe PCR if I have the primers for this disease. So that could help me with Bacillus anthracis. What if, however, you create, so this in my scenario here, I'm creating a scenario where there was farm. So let's say that that's not the situation. What if it wasn't a farm-based situation? Instead, our patient shows up with a skin infection of some kind. And now we ask them, what environment have you been in? What's your lifestyle been like and whatever? And they say, I'm pretty much just a stay-at-home person, though I did have to go to the hospital recently. And this skin infection that I have is basically a localized boil that is red, hot to the touch. The patient also has a fever. For the redness, it doesn't seem to be spreading. It does seem to be maintained as localized. All right, so now what kind of lab test might we do to identify this? So now I'm more likely to suspect something like MRSA. So one thing that I might do is a Kirby-Bauer test to see what our antibiotic choices might be. I might do a gram stain. This is a gram positive. MRSA is Staphylococcus aureus. We're looking for cocci that are organized in clusters. So that could tell me some information. Another thing that I could do to diagnose MRSA is to grow this on mannitol salts agar, where we learned in lab that this would turn yellow because it fermented mannitol. A lot of you guys got that one wrong. Not lactose. There's no lactose in this. All right, so it fermented mannitol and it was able to do that and turn it yellow. And it grows because of the high salt and is gram positive. Well, this is not surprising for something that grows on the skin. So we could do some stuff like this. There also might be some rapid tests or some PCR. And again, we would need primers for Staphylococcus aureus in order to test for that. So there's some tests there. We might culture from the skin as well on that mannitol salts agar. That's a culturing step that we would have, swabbing the wound for that sort of situation. And then we have what general medications might you offer for treatment? We're going to reach for antibiotics. I would probably select an antibiotic that affects the cell wall if I can, because this is a gram positive and our gram positives have a thick cell wall that they need to have in order to survive the salt. And so we want to do a cell wall targeting antibiotic. And so if it's already resistant to methicillin, and you guys don't need to know this necessarily off of your head, but that's why we would reach for vancomycin for this one, because vancomycin is a stronger antibiotic than methicillin, and it still targets the cell wall. So vancomycin would be my choice here. And you might not have gotten to vancomycin, but you should have at least been able to get to a cell wall because it's a gram positive. What suggestions might you have to the patient to help them prevent this from happening again? Be sure that anytime you're visiting the hospital, that you are sanitizing your hands before you leave, that you're washing your hands. Any of the surfaces that you touched could potentially have any of these bacteria on them, as well as potentially even endospores from C. diff and things like that. So be sure that you're washing your hands every time that you leave the hospital. Especially if you have any open wounds like nicks from shaving or something like that, where the bacteria could have gotten in. So what other suggestions might we have other than that? Hygiene is probably a good one. And if you do have any open wounds, be sure that you are cleaning them appropriately as well. So we might choose something like alcohol or even, what else? Alcohol or maybe potentially hydrogen peroxide or something like that. Now, we don't necessarily have to remember this, but now we're starting to get into this idea of microbial control. So how might we control MRSA? Well, it's gram positive, so we might target the cell wall. Choosing something that uses osmotic pressure might not be as useful because they can survive high salty concentrations. So salt isn't really going to work well for controlling these guys. These guys also happen to be aerobic. They do produce catalase. So I would not use hydrogen peroxide for these guys because they will be able to fight it off. If we had a high enough concentration of hydrogen peroxide, we could even sterilize areas with that. So that's going to be 35% or higher. And we usually are going to use that in a vapor format. We're going to be doing that. So we could be doing that. We could also use mechanical control of these or physical. We could autoclave any instruments that touched any MRSA to sterilize. If we boil things, this would still be good because this guy doesn't have endospores, which is the problem with boiling. We could even wash our hands The detergent in the hand wash will allow for physical removal of those microbes. I'm literally scrubbing them away. That physical action of that would help remove them. The detergent in there also, whenever we are talking about detergents, we will be talking about surfactants, including our quaternary ammonia compounds. These will act to suds for physical removal. As well as the scrubbing action, as well as it will weaken the membranes. So these guys do still have cell membranes of our bacteria as well. These will be a little bit more resistant because the wall can still help hold it together. So the weakening of the membranes isn't as important as the physical removal here, but it could work for that. If we were to use alcohols, remember that alcohols, let's say we use 70% isopropyl. If the alcohol disrupts the walls and the membranes, wow. And the water in the alcohol causes the denaturation of the proteins of the bacteria. That will also lead to killing the bacteria. It might not sterilize the area of any endospore molecules, but it should be able to get rid of bacteria like this, where we have actual vegetative bacteria. And most of our cleaning methods will get rid of our vegetative cells pretty well. All right. So skin infection for that one, we could repeat this with a respiratory infection and approach it from the respiratory side of things, whether we're talking the control methods, or we could even be talking about other things that are involved in controlling that medication-wise, presentation of the disease, how we would test it, and everything like this. But this was just for skin infections here. All right, so I'd recommend that you guys be able to go through that and explain everything that you think is involved in the things that may have led to causing those diseases: respiratory, cardiovascular, lymphatic, GI. If we were to approach this in a more specific situation, if we were to say this is a hemorrhagic disease, and so you might ask your patient more epidemiological questions like, where have you traveled recently, and how much time you spent there, how much time you spent around the people there or the animals there, or whatever, depending on what type of disease they have. Have you been bitten by a tick or a mosquito lately, and that sort of stuff. All right. Do you guys have any specific other questions for me? So we've gone through a couple of things and kind of framed basically how I expect you guys to go through this stuff. Do you guys have any questions on any of the content for me? All right. Well, if we don't have any other questions about any of this, then I would highly recommend that you guys take the time to go through these, talking about what happens whenever you get exposed to pathogens, for viral infections, for bacterial infections, and such. We kind of went through HIV on this one, and so it's a little more complicated because we do affect the immune system with that actual viral infection, and that's what leads to the killing of our CD4 positive T cells by our CD8 T cells, and then all of our infected CD4 positive T cells get killed off. We don't have any captains, and now suddenly you are susceptible to those opportunistic infections, and that's what leads to the presentation of AIDS, which is an acquired immunodeficiency syndrome, which is a secondary hyposensitivity, because it is a hyposensitivity that we got later on in our life, and we weren't born with it like we have with our primary immunodeficiencies or hyposensitivities. So our immunodeficiencies that we're born with, things like SCID, which is severe combined immunodeficiency syndrome, that's due to a genetic defect, something that was present from birth. And so people like that also would not be able to, respond well to that, to infections in general. And the worst type of immunodeficiencies are the types that affect your T cells, because your T cells are your captains and basically control the B cell response. Having immunoglobulin deficiencies from B cell deficiencies are going to be less severe because your T cells can still navigate through inside types of infections and still have a proper CD8 T cell response with the cytotoxic response. So when we talk about cytotoxic responses, there's basically your CD8 positive T cells that will be going around. That CD8 recognizes MHC1. MHC1 presents inside antigen on any nucleated cell and that is infected inside with something that will activate our CD8 T cells if we get the appropriate response from our Th1 cell, from our CD4 cell getting activated from the outside version of that antigen. And that will drive our Th1 captains to turn on our CD8 positive T cell response, then leading to expression of those granulocytes, not granulocytes, excuse me, the granzymes and the perforins, leading to apoptosis of those targeted self cells that are infected on the inside with whatever your pathogen is. So yes, so I'm going in a roundabout way, coming back to things, circling around. Yes, we start talking talking about immunodeficiency, and we start talking about the different types of immunodeficiency and why the CD4s are important, and then what happens with the CD8s, and you should still be able to have the CD8s. But you just can't make immunoglobulin, no matter how hard you would try to do that if you have a B cell deficiency, but you could still have the CD8 T cell response. But yes, if you guys can take topics like this and literally do this, just cycle back to as many things as you can. Okay, so if we're talking about MRSA, now we're talking about the Kirby-Bauer assay. What's the Kirby-Bauer assay? The Kirby-Bauer assay is where we take agar, and we might have our patient sample on the surface of the agar, and we're going to put discs with antibiotics on them. And so basically based on how big the area of inhibition, the zone of inhibition is, where we have no growth, and that is going to be more sensitive the bigger that area is, and the smaller it is, the less sensitive. And it's inverse for the resistance of that antibiotic. And with that zone of inhibition, the larger that area is, you can actually use the edge of the zone of inhibition to determine the minimum inhibitory concentration of what would be needed to inhibit growth of that bacteria. And you want to deliver the smallest amount of a drug to a patient to avoid the toxicity. The ratio of the difference between where a drug is toxic to a person and where it's effective, the minimum inhibitory concentration, where it's effective against a pathogen, that ratio is called the therapeutic index. The difference between the two is called the therapeutic window. The idea that we want a drug that is going to be effective against a pathogen but isn't going to be toxic to the person, that idea is called selective toxicity. And whenever we're choosing our drugs, we want drugs that are going to be the least toxic possible to our patients, especially if our patients have already pre-existing conditions like kidney disease or liver disease, which is going to be the primary part of our body that deals with those drugs. And so if we want a drug that's going to be very harmful to the pathogen, but not going to harm the host. And if we're targeting a bacteria, one good thing to target is peptidoglycan, because we as eukaryotes do not have peptidoglycan cell walls. And so our cell wall inhibiting antibiotics are going to be choice because we don't have peptidoglycan. So inhibiting peptidoglycan protects the patient from any toxicity inherent within that drug itself. And so things like penicillin, vancomycin, bacitracin, and many of the other cillins are going to be great. Cephalosporin and carbapenems as well, all of our cell wall targeting antibiotics. The cillins and cephalosporins and carbapenem are all beta-lactam drugs, and beta-lactam drugs are a class of drug that have a specific shape in their chemical structure that is for blocking the peptidoglycan cell wall synthesis of these bacteria. So the way that peptidoglycan works is we have a whole chain of sugars that are linked across with peptide bridges, and that creates that scaffolding structure of peptidoglycan. And this is why gram-positives that have a thick peptidoglycan wall have many, many, many layers of that scaffolding that traps crystal violet, and that crystal violet is locked in place by the mordant iodine, and that when we rinse it with alcohol, ethyl alcohol after that, then that would remove any unfixed crystal violet from a gram negative, but the gram positives, it cross-links it in place in that layer upon layer of our peptidoglycan, locks that crystal violet in place, and then they stay purple after the gram stain, whereas the gram negatives are red. I hope that I drug that through enough topics there, but you get what I'm saying here. So let it lead you away from the question. Follow the information as far as you can. Telling me as much as you can. If you get to a place where you get stuck, then go revisit your notes, go back and explain it, but use these questions as a way to lead you outside of the question. Tell me as much as you can from as many of the topics of the course as you can, and have your study sheets there and your notes there too to help you when you get stuck. But if you can't figure out when you get stuck, why you're stuck, or how to figure that out, or how it relates to our information, then that opens up an idea of some of the topics that you might not understand as well. So I just sat here and talked through all of this stuff because I have a good understanding of it. Some of this stuff I wrote down for you guys, but I can talk through it without having to look up a note, and because I have learned it and I understand it well enough that whenever you guys ask questions about some of this stuff, oh, why does the crystal violet get stuck in the gram positives and not in the gram negatives? And I can be able to explain that to you in words without having to refer to a diagram or recall some note that I saw somewhere or anything like that. I can just recall it from my own understanding of that content. And I know that's a lot to ask at this point, but that is exactly what the goal should be. And definitely work the hardest on the topics that you already know that you're having trouble with. So yes. All right. So if you guys don't have any specific questions for me, I'm going to go ahead and cut it off here. I will be posting this stuff for you guys on Canvas. So I'll post this video in whatever notes I made. I don't know how helpful they're going to be, but I'll post them anyways. And so thank you guys for joining. Thank you guys for coming today. And if you guys are studying and you get to any stuck points, then please let me know and I'll be happy to help you guys solve through that stuff. Otherwise, thank you guys so much, and I will see you guys later. I'm trying to. Yep, no problem. All right.